meiosis. So meiosis is going to be the process that creates gametes, right? It's going to be the basis of sexual reproduction because when you create gametes, those gametes fuse and they create a genetically unique individual. So let's look at the cycle here. Meiosis is going to create our egg and sperm. Remember that in the adult person, all of our cells will be diploid, 2N or 46 chromosomes. But we need to make gametes that have half that number of chromosomes. So the process that does that is called meiosis. Meiosis is a process that happens only in specific cells that are fated to become egg or sperm, and they are going to not only half the number of chromosomes, but they're also going to recombine the DNA within the mom and also within the dad and create basically gametes that are genetically unique from each other. So every egg the woman makes in her life is unique, and every sperm the man makes in his life is unique. So meiosis is this process. We get our gametes. When they fertilize, we create a zygote. And the zygote is going to be our 2Ns. This is the first cell that a person will arise from. And then this diploid zygote will undergo many, many mitosis reproductions and create the person. All right. So this is another concept check. You can answer this. And let's take a look at the phases of meiosis. So be aware that meiosis has two phases to it. There's meiosis 1 and there's meiosis 2. Meiosis 1 is very different than mitosis. Meiosis 2 is identical to mitosis. So if you understand mitosis, you already understand meiosis 2. Meiosis 1, there are some little changes, and I'm going to go through them. Okay, so meiosis 1. So notice we're going to use the number 1. So if you look at your outline, you always want to put a 1 in front of meiosis 1. So it's prophase 1, metaphase 1, anaphase 1, telophase 1. What you're looking for is, what we're looking at here in this image, is that this is our homologous pair. So we're going to say the red one is the maternal chromosome, and the blue one is the paternal chromosome. So these are homologs, right? Now, we know that each one of our chromosomes is going to get copied in S phase of interphase, right? So we have the copy of mom's chromosome, the copy of dad's chromosome, and just like mitosis, these sister chromatids will be attached to each other at the centromere. Now, what's unique about meiosis, what's unique about meiosis is that the homologs, the mom and dad's homologous set, will come together, and then they're going to switch parts. We're going to take a look at that. In meiosis 1, the whole thrust of meiosis 1 is to separate the homologous pairs. Okay, because ultimately what we want are gametes with just one homolog. Meiosis 1 does not separate the sister chromatids. That's what meiosis 2 is for. So meiosis 2 will separate the sister chromatids into those separate cells. All right, so again, meiosis 1 is to separate the homologous pairs. Meiosis 2 is to separate the sister chromatids. Now, this is kind of an interesting phenomenon. After meiosis 1, this cell, if a cell has both homologs within the same cell, so you have mom's chromosome 1 and dad's chromosome 1, this is a diploid cell. If you separate the homologs, right, mom's chromosome 1's over here now in another cell, and dad's is over here, technically this cell is haploid, because the homolog is no longer found within the cell. Yes, the chromosomes duplicated, we have two sister chromatids, but it's still considered haploid because the homolog is not there. Okay, and so this haploid cell is going to separate the sister chromatids. All right, so let's take a look at the events of prophase 1. Now, interphase is the same Nothing changes in interphase. We have our centriole pair or our centrosomes. We have our chromatin. The DNA is getting duplicated. In prophase 1, this is the very unique phase of meiosis. As I mentioned before, the homologous pairs will find each other during during prophase 1. This does not happen in mitosis. So let's call the longer chromosome here chromosome 1, and we'll call the shorter chromosome here chromosome 2. So we can see that we have maternal or mom's chromosome 1 and and dad's chromosome 1, right? They're homologs. They are creating this unit. They find each other in prophase 1 and they create a tetrad. So if you look at letter A under prophase 1, I mentioned the homologous chromosomes pair up. This makes a unit of 4 chromosomes called a tetrad, right? So this unit here is the tetrad. Here's another tetrad. Now, let's look at letter B. Crossing over occurs between the homologous chromosomes within the tetrad. Crossing over creates recombinant chromosomes, which creates variation. So if we look at our 2 center chromosomes, right, we have dads and moms, they're crisscrossing, right? There's a little crisscross going on here. When they cross over like this, they physically break off the parts and swap parts with each other. And so if you can imagine, this inner chromosome 1 on mom's side will have a tip that is blue. So it'll have a paternal region. All the rest will be mom. And then down here where they switched again, the rest of that mom's chromosome will now be swapped out with dad's. Because we have this mixing of chromosomal information, the resulting sister chromatid, right, mom's, this one chromosome, is going to be recombined. So we call that a recombinant chromosome. All right, so hopefully that makes sense. We're crossing over, switching parts, but just in the middle. So those outer chromosomes on the edges do not cross over. Just the 2 in the center of our tetrad. Okay, that's the big deal. So that is very special in prophase 1 in meiosis, the crossing over. And to have crossing over happen, you have to have the tetrads come together. All right, so then metaphase 1 is exactly the same. The centrosomes are going to find opposite poles. The spindle fibers will grow. They're going to attach to the centromere of our chromosomes, and they're going to push our tetrads, okay? They're going to push that unit of 4 along the metaphase plate. So, what happens next? Anaphase 1 separates our homologs, okay? So we're shortening those spindle fibers. We're moving one homolog to one side and the other homolog to the other side, right? So let's go back. Remember I mentioned this is the thrust of meiosis 1. We have the set of 4. We're separating the homologs into different cells. So that's what's happening in anaphase 1. Now, notice that the little unit here, there's one chromosome, this chromosome 1 that's blue. It's completely blue. That was that original chromosome 1 from the dad. But the other chromosome from the dad, the other sister chromatid here, right, it is different now. It's been recombined, so we have a recombinant chromosome. On the mom side, we still have an unaffected or unchanged chromosome 1 from the mom, but we also see that we have a recombined chromosome 1. So, and the same for our second chromosome here. So just keep that in mind. We're still separating the homologs, right? So we're still separating mom and dad's homologous pair, but just that there is a little bit of a change in some of our chromosomes. Telophase and cytokinesis happen. This is a normal, nothing special happens here. And then we're going to move on to prophase 2. So you need to remember that there's no interphase between meiosis 1 and meiosis 2. So just once telophase 1 happens, it just jumps into prophase 2. So prophase 2 is, and metaphase 2, anaphase 2, that's exactly like mitosis, right? So we have our sister chromatids attach to each other. We are breaking down our nuclear envelope. We are having our centrosomes moving to opposite ends of the pole. It's showing you two cells, right? Because in the meiosis 1, our one cell became 2. So in meiosis 2, we have 2 cells undergoing meiosis 2. Metaphase 2 is exactly the same as mitosis. Anaphase 2, again, exactly the same as mitosis, and then telophase 2. So the thing to remember now is in telophase 2 at the end, you're creating 4, right? 1, 2, 3, 4, 4 haploid daughter cells. And if you look at the combinations of the genetics inside each one of them, that no two of them are the same. They're all genetically different from each other, which is a really big thing to understand. That if you look at your outline under number 4, telophase 2, you get 4 haploid cells made. All of them are genetically different from each other. Okay, so that is the key in meiosis, that every single one of your sperm and every single one of your eggs that you make throughout your whole life is going to be different genetically from each other. Okay, this is just a nice picture of meiosis 2 in a lily cell. And then here's another concept check for you. And let's look at how genetic variability, letter C in your outline, is created. So why does the process of meiosis have crossing over and building in variation? Because it's healthy for a population. We need to make sure that we are genetically different from our parents. And let's talk about why. So let's assume that all people on the planet were identical. We're all clones. Then if somebody is going to fall ill, COVID-19, for example, right? If someone is very, very susceptible to COVID-19, that means that if they fall ill and die because of that infection, and everybody is genetically the same, everybody will have the same weaknesses, then everybody will die. That's not healthy for the human population, right? So it's healthy for any population of any species to be different, genetically different from each other, so we all have different strengths and different weaknesses. So that when you have turbulence and disturbance and different kinds of pathogens that arise in the world, that there will be some individuals that are going to survive and some that maybe won't, but because of the variation, you'll always have some survivors. So the 2 ways here of making variation is the recombinant chromosomes, as we saw in crossing over. And then number 2 is the independent assortment of chromosomes. So let's talk about this. This is an image, again, of the crossing over, right? And then what we are creating in the result is recombinant chromatids, or recombinant chromosomes. But this is independent assortment of chromosomes. This is something that you need to think about. So what independent assortment of chromosomes mean is that when your tetrads, or when your tetrads align along the metaphase plate, they can do it randomly. So in this scenario, you can have mom's chromosomes lining up on the same side and dad's on the other side, or just as likely, another possibility is you'd have them on opposite sides, okay? Just a different way. So that means that the chromosomes are assorting themselves independently, that chromosome 1 lines up independent of what chromosome 2 is doing. It doesn't matter what chromosome 2 is doing, right? So they're kind of just doing their own thing, aligning themselves along the metaphase plate. If you go through the motions, you'll find that under possibility 1, these are the gametes you get. Under possibility 2, these are the gametes you get. So just looking at two tetrads, right, and how they line up along the metaphase plate, you get different combinations of gametes at the end. But most things have more than 2 chromosomes. This is you don't need to worry about the math here, but if you're thinking about we have 23 different chromosomes, then how are all the different ways? There's possibility 1, possibility too, but these are only showing 2, right? We have 23. So how many possible ways can we arrange the chromosomes facing all the different directions, like all the possibilities? There's 8 million possible combinations. All right, that's a lot. So the resulting egg and the resulting sperm is basically 1 out of 8 million possible ways, possible combinations, right? So an egg may be fertilized at random by 1 sperm cell. That's random fertilization. An ovum is 1 of approximately 8 million possible chromosome combinations, and the sperm represents 1 out of 8 million possible combinations. The resulting zygote is composed of 1 in 64 trillion possible combinations of chromosomes. So that means that your mom and your dad, right, they could have the possible number or the possible different kinds of eggs that a mom can make is 64 trillion. Just because of meiosis and the way that you have this independent assortment and you have the crossing over, there's 64 trillion possible eggs, right, that your mom can make. There's 64 trillion possible I'm sorry, there's 8 million possible eggs, 8 million possible sperm. There are 64 trillion possibilities when you create the zygote. So basically you are 1 out of 64 trillion, right? So if your mom and your dad had 64 trillion babies, you might get reproduced once, right? So that's pretty remarkable. All right, so is sexual reproduction worth it? The process of meiosis is very energy consuming. Remember, it's 2 steps, 2 phases. Finding a mate is very difficult for organisms on the planet, even for us, right? Finding a mate can be difficult. Asexual reproduction is great for loners. If all you need to do is divide yourself in half and to increase your population, then that's the easier route to go. However, why would all of the animals pretty much reproduce sexually? There must be some advantage, right? And that was what I was that's what I was talking about earlier with COVID-19, is that to have sexual reproduction means that you're ensuring your offspring is different genetically, and that having genetically different individuals in a population is very healthy for that population. If you look at 4 organisms that can reproduce asexually or sexually, would you expect them to have sexual reproduction during stable times or unstable, stressful times? So there are some organisms out there that can either asexually make a clone or find a partner and create a unique offspring. When would you expect them to have sexual reproduction? Stable or unstable times? And it turns out a lot of people say stable times because it's costly energetically, and you have to find a mate, but actually it's unstable, stressful times. So if you feel like the environment is bad, it's stressful, you might not be able to survive it, then wouldn't it be good to have offspring that are different than you so that maybe they can survive the stress if you don't? Right? If you made a clone and you're stressed out, then your clone's going to be stressed out, and that's not good for the population. So anyways, just some things to think about. And here's our last one more concept check for you. And I will look, so the last thing here to talk about is nondisjunction. So nondisjunction is a mistake in meiosis. It can happen in meiosis 1 or it could happen in meiosis 2. Basically what happens is either the tetrads, the homologous pairs, fail to separate in meiosis 1, or the sister chromatids fail to separate in meiosis 2. Either way, what you're going to get is an abnormal number of chromosomes, and that's the thing to remember. Nondisjunction can occur in meiosis 1 or meiosis 2, but what you're going to get is an abnormal number of chromosomes. You either have an extra chromosome or you could be missing a chromosome. So this is what happens, right? You have an abnormal egg with an extra chromosome, a normal sperm cell, but then you get 3 abnormal number of chromosomes in that zygote. As we mentioned before, trisomy 21 is a result of nondisjunction, right? You have an abnormal number of chromosomes. You have 3 of chromosome 21. That's not normal. So Down syndrome is known to be increased with the age of the mother. And there is a lot of physical and mental traits that go with a Down's baby. And so before we leave here, I just wanted to, I thought I had a graph. I mean, yes. So this is the graph of the age of the mother and the incidence of Down syndrome. So you can see that the older the mother is, the higher the rate of Down syndrome. So there must be something with the health of the eggs when they undergo meiosis further on in age, that the separation of either the homologous pair or the separation of the sister chromatids don't occur in a regular manner. Okay, so let's go back. This is your number 10 concept check. And let's wrap up the lecture here. So the examples of nondisjunction, there's another one called Klinefelters where you have an extra sex chromosome. You can see you can have lots of extra sex chromosomes, and you can read about what the Klinefelters is about. And then these are just some examples of nondisjunction in sex chromosomes. So different syndromes. I do not expect you guys to memorize any of these. And then you can fill in this chart of mitosis and meiosis, and I'll save this for another wrap-up video. But these, I'm going to just scroll through these so you can answer these. You can pause the video and answer these. So this is 11 and 12, 13 and 14, 15 and 16. Okay, so submit those answers to me. If they're correct, you'll get extra credit.